Hi. So far we've talked about DNA. Now we turn some attention to proteins. What you're looking at here is from a paper by Tyson et al. The amino acid length on an average of proteins from multiple species. You see on the purple side, Placozoa, Diplomonalia, etc. Then you see vertebrates. Blue dashed line indicates humans. We stand at an average of about 500 amino acids per protein, whereas bacteria are at 300 amino acids per protein. Okay? So this means that knowing these statistics, by the way, these statistics are publicly available on PubMed and the protein databases also, we can potentially also make sense of the protein abundance. In a sort of similar spirit that I had shown earlier, which was missing the distribution of plant proteins, here you're looking at average length of plant proteins in terms of amino acids. And the average value again is somewhere around 200 to 300 amino acids for plant. The best model for this fit is a log normal from green algae chlorophyta. So the XPC database protein scale also tells us about the molecular weight of amino acids. This is centered around 130 in terms of mass in daltons, which means that we can allow ourselves to approximate mass of an amino acid as 100 daltons. So combined with the length amino acid as 300 and mass per amino acid as 100, we end up with the mass per protein on an average of 3 into 10 to the power 4 or 30 kilodaltons. Now, I've made many approximations and simplifications here, but I will proceed to show you that these are quite useful. This is not a typical protein, which is why I put it in inverted commas, but it's a protein you should know about. The discovery of the protein itself is attributed to Osamu Shimomura, who won a Nobel Prize for its discovery in chemistry. As you know, there's no biology Nobel Prize. It's for medicine, right? So you can claim many biologists win that, and many biologists also win the chemistry Nobel Prize. Sometimes they even win, but not. Sometimes they may win a physics Nobel Prize. The proteins per E. coli is the question we want to ask. So one way to get at it is to ask what is the volume of a cell. So we recall that we said that the cell is a rod of 1 micrometer or spherocylinder 1 micrometer height or diameter and length of 2 micrometer. And if we ignore the semispheres here, then the volume is pi r square h, which is, again using my calculator, I get something in the order of r square, which is 0.5. So 0.5 squared is 0.25 micron square into pi is 0.78, that's So 3.8 into height is 2 microns. So I'm left with 1.5, and this is micrometer cube. The dry mass of a cell is 30%, which means 70% is water, which we have discussed earlier. Proteins form 50% of the dry mass, and 2/3 of the proteins are cytosolic proteins. The remaining proteins are membrane proteins. So the idea is that if you know the mass, which means from the volume, we get the mass of a cell as the density by volume, which is 1 gram per cm cube by 1.5 micrometer cube, which is 10 to the power 3 kg per meter cube upon 1.5 into 10 to the power minus 6 cubed is minus 18 meter cube. This leaves us with, so you can use your calculator now. So the exact number is a very strange one. It's 666 into, so 6.6 into 10 to the power so I hope some of you have noticed that there's a mistake here. We are expecting 1 picogram, with the mass of the cell being the mass of the cell being only part of the picture. We want to multiply it by 0.3 to compensate for dry mass, and by 0.5 for the protein mass, and in addition by 2/3 for the cytoplasmic food. All this divided by mass per protein will expectedly give us the number of proteins. You can do this calculation yourself and check if these numbers make sense, because we expect 10 to the power 6 order of magnitude proteins per E. coli cell. Now, if we ask the next question, which is how full is the cell with these proteins? Then we're asking a question about protein spacing. The variables that we need are the number of proteins, which we just identified as 10 to the power 6, the volume of an E. coli cell, which is a femtoliter, and the density, which is the number of proteins per volume of coli. That will give us the volume occupied by a single protein. But we want the spacing. So using dimensions and simple numerical literacy of what these mean, if we take the volume per number of protein and take the cube root of it, then we in fact have a distance in dimensions of L and units of meters or micrometers or nanometers of 10 to the power minus 18 meter cube, because of the volume of 10 nanometer distance. Now, it's important to bear in mind, and that's why we brought up this protein here, that a single protein, like a green fluorescent protein, which is found in a jellyfish, has a side length of 4 nanometers in terms of the height of the barrel, and a diameter of 3 nanometers. So roughly a few nanometers, 3 to 4 nanometers. That is what we argue is the size of a typical protein. The spacing between the proteins is 10 nanometers. Assuming that each protein is only a point particle, the protein to protein spacing is 10 nanometers. If, however, each protein has a diameter of 4 nanometers, then it has a radius of 2 nanometers. You take out 2 from each side, so you're left with only 6 nanometers. This is roughly the size of another protein. In other words, the spacing between proteins, assuming only proteins populate the cell, which of course is not true, is that of one protein. In other words, it's very hard to move around for proteins because there are the additional structures like ribosomes, DNA, small ions, and all of these serve to provide the crowded environment inside the cell. So I want to now switch to a model gene which can function as E. coli cell functions as a hydrogen atom of a cell, as a hydrogen atom of a gene. That gene is the lac operon.